Good afternoon, good morning, good evening, wherever you are. Thank you very much to the organizers for inviting me to share a bit on rapid pathogen identification from blood cultures, and also some preliminary results from a randomized control trial that we did in Singapore. My disclosures: I have received in-kind support for the work discussed in this talk from bioMérieux and BioFire, and also ROSCO Diagnostica, and some of the work that we'll present was supported by a generous grant from the National Healthcare Group for the Clinician Scientist Career Scheme. Today we'll cover, I think some of it was alluded to in the previous talks, overview on rapid diagnostics for pathogen and resistance in 2021, focusing on bloodstream infections, which have the highest morbidity and mortality, and the value of RCTs in diagnostics, in particular BSIs, and also some results from this trial called the RABBIT trial, which we are wrapping up. So the burden of bloodstream infections for bacterial range around 113 to 204 per 100,000, depending on where you are. And the table on the right, I think you can see that there have been estimates in different time periods in different places in the world. I think what's interesting is that not only that they form quite a substantial burden, this burden is similar to stroke or the rates of DVT, a bit lower than heart failure, but it's much higher, for example, than type 1 diabetes or even suicide. So this is a substantial burden. And if we look at the Sentry data, which has looked across multiple countries from all over the world, we can see that over the last decade or so, the rates of antimicrobial resistance in gram-negatives have increased. A lot of it is driven by ESBLs, the extended spectrum beta-lactamases, MCs or the cephalosporinases. There's increasing fluoroquinolone resistance, large part driven also by use in agriculture. And there are some, besides resistance determinants, there are successful clones like E. coli ST131, which have spread worldwide. In terms of their effect on delay in effective treatment, we know that with resistance, if you start empiric therapy and it is wrong or it is ineffective, there's a delay in patients getting the necessary antibiotics that they need. And in terms of mortality, that's where the data is a bit kind of back and forth here. There's some data that maybe between organisms, Clap pneumo seems to have higher mortality rates if you give ineffective antibiotics compared to E. coli. Some studies have found no real difference in mortality, and I think that there might be region and also species differences when it comes to mortality outcomes. Now, we were interested to look at Singapore and another geographically diverse area. So we worked with collaborators with Dr. Patel from the Mayo Clinic, and we looked between October 2013 and March 2014 and looked at E. coli from two very different geographic areas. I think if you have been to Rochester, Minnesota, it's a very a nice town, a quiet town in the Midwest. They do see quite a bit of medical tourism with folks coming in, but by and large, it's quite sheltered, and for half the year, it's quite cold. So they do see lower rates of antimicrobial resistance here compared to Singapore, where in the heyday prior to COVID, we had more than 20 million visitors a year and to Singapore. And we see a lot more traffic and also higher rates of antimicrobial resistance. Interestingly, the successful clone ST131 E. coli accounted for about a quarter. So as a clone, it's pretty successful in both at both sites. However, within ST131, there is a clade known as the H30RX clade, which was more prominent in Singapore than Minnesota. So this was 15% in Singapore versus 4% in Minnesota. And when we looked at that, patients in Singapore with this, if you had ST131 E. coli infection, bloodstream infection, you are more likely to receive inactive initial antibiotics about 3 times. And especially if you had H30RX, which is the ESBL clone, this went up to 7 times. What was concerning to us was that about 16% of community onset bacteremia episodes in Singapore, but none in Minnesota, were actually from the H30RX clone. So I think some of this was covered in the 2 preceding talks. Now we have a number of FDA approved rapid blood culture diagnostic assays. I think here pictured the 4 which are more commonly that we see in the market, and then there's the ePlex in this slide where they have BCID, also gram-positive, gram-negative, and a fungal panel with expanded targets on it. So some of these panels, the chemistry, detection chemistry is different. Some of it, for example, T2 uses magnetic resonance measurement. For the Verigene uses gold nanoparticles and detects the nucleic acid targets. BioFire is a nested, is a multiplex PCR in individualized wells, and it's a closed system. Accelerate Pheno, as was discussed, is a phenotypic test with real-time microscopy and fish probes to detect the organism. So all these are all really, really exciting. And for the BioFire film array, they have recently upgraded their panel from a version 1, which we did our study on with the version 1 panel, to a version 2 panel, which has expanded antimicrobial resistance gene targets, yeast targets, and more bacteria. This just also gives you an idea here. This is the Accelerate panel and the time, the targets that they can detect in terms of both gram-positives and gram-negatives, and also the antimicrobial agents which are included in this panel. So just in summary, I think those were busy slides, but looking at all these panels, I think it's important to note some of them, the clock starts ticking from the positive blood cultures. So you need to have still take blood cultures and still incubate, and then when the instrument flags positive, you pull it out, and then you get that turnaround time, which for a lot of the panels is about 1 to 2 hours. And then for the only panel which has phenotypic AST for now, which is the Accelerate Pheno, AST results are available in about 7 hours. Some of them direct from blood, but for example, the T2 panel for Candida and bacteria, but the number of targets are more limited in those panels now. And then it's also direct from blood, so you're not really sure also. I think stewardship would, like who do you use such a technology on, and how do you risk stratify to kind of decide which patient gets which, gets the test. Those might be challenges because it's going to be drawn directly from blood. And some of these technologies, as was mentioned before, were a bit more expensive. I think what's also emerging here is not just the accuracy of the test, but also real-world practical outcomes. So as you can tell, some of the resistance patterns, prescribing patterns, so doctors, generally, I think physicians, having worked away, tend to behave in mostly similar ways. Thankfully, most of the guidelines that we have and ID physicians all over the world tend to have similar prescribing practices, but there are going to be regional differences. So some will be more cautious, some will be more gung-ho, some would be steward maybe better in some ways, or maybe too conservative in someone else's opinion. So, and also the bugs, as we have shown, also have regionally there are differences in resistance rates. So beyond the accuracy for each test as to the correct organism identification and also resistance detection, what really is the clinical impact when contextualized to antimicrobial stewardship, and also the specific antibiotics that choices which are made thereafter. So the Antimicrobial Resistance Leadership Group from the NIH, supported by the NIH, has come up with interesting outcome analysis called the DORMAT analysis. So it is what we call real world assessment, and it is codified into a score, and it's illustrated in the clinical scenario on the right. So for example, if you had 3 rapid tests, and in this recently published study in September 2021 in CID, they compared the Veregene panel to the BioFire BCID version 1 and the version 2, which have different targets. A lot of it overlaps, but there are some different targets. And then they get several clinicians to opine on the choice of antibiotic they would choose based on the results that are obtained. So you can see that step 2, the clinicians, depending on the panel result, for Veregene, they might have chosen ceftriaxone when they got an E. coli and a negative CTXM result. For BioFire BCID, because it did not have gram-negative resistance targets, the clinicians chose piptazo, and then in the BCID2 they chose ceftriaxone. And then based on a reference panel of the score and credit, depending on when the final susceptibility results come out, if you have organism which is completely susceptible versus one which is very resistant, you can see in the matrix they are scored as to whether you get full credit, partial credit, or no credit. And finally you get the DORMAT score. And this is a practical score to see how discriminatory and how effective it is for real-world ASP. So in this study, and they looked at these 3 assays, they found that the BioFire BCID2 panel had the highest DORMAT scores. Certainly, this has to be taken in context to where the study was performed and prescribing practices, but I think hopefully more of such real-world practical scores can be used in addition to the accuracy studies that we are more familiar with. So again, here is the same group which looked at both gram-negative samples and gram-positive samples using the DORMAT approach. The full acronym is Desirability of Outcome Ranking Approach for the Management of Antibiotic Therapy. And here again, they found that the EPLEX panel, here they look at slightly different panels, the EPLEX BCID panel and the Veregene panel, and they found that while the accuracy was very high for both the Veregene and the EPLEX panels, you can see it's 98% for the positive predictive agreement. But the EPLEX DORMAT score was higher because it was able to identify more organisms and targets in the Veregene panel. So we are used, I think, in especially with the advent of COVID, a lot of RCTs kind of proving the effectiveness of vaccines and medicines. But what is not so much talked about is the role of RCTs in diagnostics. I think we're used to it, but there's also in drugs and vaccines, but there's utility also in diagnostics because the performance of the test, whether it's alone or in combination with other tests, can actually impact real-world outcomes. And here we're looking at the effect of diagnostics on actual patient outcomes. When diagnostics are applied to RCTs, these are usually called test plus treatment RCTs. And in the realm of diagnostics, some of these tests, if we were to group them, these tests could be replacement tests, treatment RCTs. They could be add-on test treatment RCTs, or they could be a triage test treatment RCTs. And the value of such trials, while not perfect, yet they have a role because they may eliminate potential bias, particularly in pre-post intervention studies. So in a lot of diagnostic studies, we do a pre-post intervention to measure outcomes, but RCTs can eliminate some of those unmeasured biases. Having said that, test treatment RCTs may also be subject to bias. So when we evaluate trials, I think some of them, they look at different outcomes for RCTs, as you can see in the table on the right, like quality of life, symptom score, adverse events, mortality, timing of care, cost. etc., etc. I think when we look at RCTs, we need to be careful and kind of be aware of how the sample size, how they allocated the treatments, whether there was blinding, the assessment of the outcomes, whether they were reported accurately, how participants flowed, how they dealt with missing data, intention to treat, and various outcome analysis. So having said that, there have been two major RCTs for blood cultures. And here, this was the first RCT. It was done around 2013 to 2014 at the Mayo Clinic. And here, they looked at the role of the BCID version 1 panel as a RCT and its real-world impact on prescribing practices and patient outcomes. In this study, they randomized 617 patients in 3 arms. There was a standard control arm and 2 intervention arms, BCID with templated comments. And then the other intervention arm had the BCID result with templated comments and antimicrobial stewardship. So one arm, you kind of left the doctors to read the report and see what they do. The second arm, you picked up the phone in addition to the report and you gave them a friendly phone call to say, hey, maybe you like to do this or that. So the results, I think as expected, BCID identified microorganisms 1.3 hours versus 22. This study found a decreased use in broad spectrum antimicrobials, piptazo, decreased treatment of contaminants, and also increased narrow spectrum antibiotic use for gram-positives. So less treatment of coag-negative staph, for example. And what was important was that there was no worsening of clinical outcomes. I think one worry is that if you get this rapid test, it may not cover everything, all the targets you wish for in a panel. And you do not have a true phenotypic result, and you're worried that the final phenotypic result you might have to re-escalate. But I think this was important that this trial showed that there was no worsening of clinical outcomes. However, of course, the context where this trial was performed was in Minnesota, where gram-negative resistance is less compared to, for example, here in Asia or in Singapore. They saw faster escalation in both intervention arms. So in Singapore, we'll say that kiasu, right? So when people read a report, we will quickly escalate. But then de-escalation, if you just gave the doctors a report to read, most of them would sit tight and not do anything unless they get that friendly phone call and someone to reassure them that yes, you can de-escalate. I think this is nicely illustrated in this diagram, where in the control arm, you can see the upright triangle, where escalation occurred later compared to the rapid arms, where it occurred at the earlier time point. You can see before about the 6-hour mark. And then de-escalation, which is illustrated by the blue arrow, when you just gave people a result and you did not have that friendly phone call, they de-escalated almost the same as if there was no rapid test. But if you threw in that phone call with the ASP team, then you brought it down to less than a day. So I think that's also a lesson in implementation of technology and human behavior. The group, similar group under the auspices of the ARLG, this was a more recent trial and published in CID in 2020, where they looked at 500 patients with specifically gram-negative BSIs in 2 centers. They had over 200 in the control arm and 200 in the rapid arm, which is using the Accelerate Pheno system plus ASP. You can see certainly with the rapid arm, there was a shorter time to results. Time to AST, 13 hours versus 44. First overall antibiotic modification, 8.6 versus 14.9. For gram-negatives, again, 17 hours versus 42. And antibiotic escalation, 18.4 versus 61.7. So we thought that we should try to replicate this, some of these studies in a context where practicing antimicrobial stewardship practices and also resistance patterns are a bit different. So I led a study called the Rapid Blood Culture Intervention Trial, or RABBIT. And in this study, which is closed and we're now in the final data analysis and cleaning up phase, And here I'm happy to share with you the trial protocol and some of our preliminary results to see whether such a strategy would lead to timelier institution of effective and optimal antibiotic therapy to improve patient outcomes, and whether we could minimize unnecessary antibiotic use, including combination therapy. Why? Because Singapore has higher rates of resistance and different practice patterns in clinical context. So the primary outcome for this trial was the time from positive blood culture to effective or optimal antibiotics. An effective antibiotic was defined in this trial to be one to which the organism was susceptible to, and the optimal antibiotic was the most narrow spectrum and targeted as recommended by the trial protocol and institutional guidelines. We are also looking at a number of secondary outcomes, including mortality. I think in a lot of trials where your mortality rates are not that high to begin with from bloodstream infections, it's hard to prove mortality benefit, as with the RAPIDS-GN trial and the other first BCID trial. But we are looking at that, including to a number of outcomes, including the antibiotic use data and also duration of hospitalization and cost effectiveness. We included patients who had a positive blood culture and gram stain who were older than 21 years old and able to consent, and excluded them if they had a positive blood culture in the previous week or had a negative blood culture bottle gram stain. Because in Singapore, at that time when we were looking at this trial, the BCID study, the first panel did not have except for the KPC target, carbapenemase, did not have other gram-negative targets, and ESBLs are an issue in this region. We decided to couple it with a phenotypic resistance test in the rapid pathogen identification arm by a company called ROSCO Diagnostica. So this is an overview of the lab process. So when the blood culture flagged positive after enrollment, so this is a prospective trial, we put that on the BCID instrument, and then for gram-negative specifically, we also did the ROSCO ESBL and carbapenemase screening kits for gram-negatives only. So this is kind of the schema of the trial. It's a one-to-one randomization. Everyone got standard of care, which is in Tan Tock Seng and NCID. The Tan Tock Seng Diagnostic Lab does actually direct from disc susceptibility. So actually, we are already getting our antimicrobial susceptibility results about 1 day faster. So it's quite unlike the previous trials in the RAPIDS-GN, the first BCID trial, that took additional 24 hours because they would work on susceptibilities from pure isolates. So I think we are putting this molecular test actually to quite a run for its money, actually, because we are comparing it to cheaper rapid from this susceptibility test, which you get 24 hours even faster ahead of time. Compared to doing it from isolate. So, and then everyone who was randomized to the diagnostic arm, the rapid diagnostic arm, in addition to standard of care, they also got the BCID panel, and then they also got, if a gram-negative, the ROSCO ESBL carbapenemase test. So just to remind us, this was the version 1 of the BCID panel, where there are 27 targets on the panel, and resistance, there was MRSA, BAN-A, BAN-B, and KPC. And maybe some of you are not so familiar with the ROSCO Diagnostica kit. It's a bit like the CARBA-NP test, but they come as tablets. And here you can see a test where it's a positive test for ESBL, where on the right, the organism is pelleted down from the blood culture, and there's an inhibitor called Tazobactam, and they use the third generation cephalosporin, cefotaxime, and it inhibits the ESBL, so the tube remains red. But the tube on the left, there's only cefotaxime, and the beta-lactam ring is hydrolyzed, and the pH has dropped, and the indicator has turned to yellow. So this indicates that there's an ESBL present. So when you are doing it though, just as a word of caution from the trial, also that there's a bit of hands-on time for the technologist. So I've listed down the steps that you have to do. So you have to take 0.5 mils of blood. If you to put it in a round bottom, it's important to use the round bottom tube so that the tablets can dissolve well. Put a bit of Triton. You have to then incubate it for 5 minutes. There's a centrifugation step. You discard the supernatant, and then you incubate it up to 1 hour. You resuspend the pellet and you wash it, and then you recentrifuge, and then you need to break the bacteria up to release the beta-lactamases. So we use a tris lysis buffer. Then you add the antibiotics and the indicators in each of the tubes, and then you incubate for 1 hour, and you look for the color change. So here are results of some of the tests. So you can see A, B, C, and D. So A is a negative test for negative, no ESBL, no beta-lactamase, no carbapenemase. This is the ESBL test. Here you see a positive test. And this is, sorry, this is, I miswrote here. This is a ROSCO Carba negative test on panel C. And then here you can see one of the tubes is actually a very weak, it's just turning orange. It was read as positive, but this was due to a weak beta-lactamase, carbapenemase, OXA48. So beyond just the Microlab results for the trial, I think it's very important to put in plain English what this means, because if you were to just text people ESBL found, KPC found, NDM found, people won't know what we don't expect our regular physicians or people who don't deal with bacteria in the lab or non-ID people to really intuitively be able to know what to do next. So as part of it, reporting is very important. So this is an example of a report we use in the study. So we say like, when is it drawn, from which bottle, E. coli detected, is positive for ESBL, and what this means. Organism is predicted to be resistant to extended spectrum cephalosporins. We don't assume that people know what an extended spectrum cephalosporin that is, so we put in brackets, for example, ceftriaxone, how we tested this, and then accompanying the result note, then there is actually a stewardship note and a phone call where we say, stop this, you must be very specific, start this, stop this. And then we even recommend some other things like imaging, and we inform the primary team. So this took place throughout the trial. I'll share some results from the study with regards to the gram-negative part of the study. So this was an interim analysis. We just published this in CID, and you can look it up. I think it's available online now, where we looked at 97 positive blood cultures. So before we introduce the test, we need to make sure the test work in a verification phase and a prospective validation phase in 123 prospective blood cultures out of the initial recruits in this trial. And what we found was that the first part, we didn't look so much at the BCID panel. We're more focusing on how the ROSCO kits perform. Out of the 54 clinical and 43 spike specimens, the ROSCO ESBL kit had a sensitivity or PPA of about 100% and NPA of 100% in what we spiked. And here you can see the ESBLs we studied, mainly the CTXM group. And the RC kit, the ROSCO carbapenemase kit, detected only about 50% of the carbapenemases we studied. I think it's important to see what carbapenemases we used in the verification phase. Some of it was spiked, as you can see. It didn't do so well with the weaker carbapenemases like the OXA group, SME, SME and VIM and IMI groups. But it worked well for KPC and NDMs. For the prospective clinical cohort group, we actually, thankfully, carbapenemase producing organisms in blood cultures are not so common. We only had one OXA48, which I showed the picture of in the prospective phase of the first 123, and the RC kit did detect it. For the BCID, for the ROSCO ESBL kit, there were 27 ESBLs. It detected 74% of the ESBLs. Amongst the ESBLs, though, in the CTXM group, it detected all. So I think the CTXM ESBLs are the most common. However, it did not do so well, and I put in the red box here some of the TAMs and the shifts. Yeah. For the BCID panel, for the on-panel target-based organisms, it picked up 99%. And if we look at just on a sample-based PPA or sensitivity, it was 99.1%. The panel only missed one Klebsiella in a polymicrobial infection. I think in the study, during the study, the manufacturer also issued alert of Proteus false positives. We had 2 Proteus false positives in this study, but those were suppressed. And I think that's another thing to note is that the blood culture bottle manufacturers are not obligated to make sure that their products are nucleic acid free. So it's important to correlate the gram stain with the results that you get read out. We did do, just as a midpoint, safety analysis for mortality. So this trial enrolled over 800 patients. In the study arm, for all patients, the mortality was 9.9%. In the control arm, 15.5. This was a 30-day mortality. It didn't reach statistical significance. Again, this was a safety signal, and we are in the midst of analyzing the full results, and we hope to be sharing those soon. Well, in conclusion, for the rabbit trial, I think what we felt so far is that the rapid diagnostic algorithm, sometimes being a bit creative and combining both molecular and phenotypic tests, allowed rapid identification for most of the gram-negative pathogens and their beta-lactamases, allowed for faster clinical decision making, and we are sharing, hope to share more results soon. And in summary, for blood culture diagnostics, it allows these rapid culture diagnostics. I think we need to continue to learn how to use and implement them and contextualize them to where we practice. But in general, they do allow more timely escalation and de-escalation of therapy. I think what's important to know is where you are practicing and what kind of resistance patterns you have at your area of practice, because this would affect your choice of technology and how you would roll it out. And more importantly, with studies and as you implement it in your institutions, besides demonstrating the accuracy, other novel measures of impact, for example, new scores such as the DORMAT trial, not just accuracy alone, I think are going to be more commonplace in the future. So I'd like to thank the collaborators in this team. I think there are many, many of them which are on this slide, some of them who also spoke in the earlier session. And also special thanks to the patients who participated, and also the laboratory staff at Tan Tock Seng DLM, and also bioMérieux for supporting this work, and ROSCO Diagnostica and NHG for supporting this work with a grant. And this is my last slide. I think in the era of COVID, we just have to remember that bacteria can make us sick, but most of the time they're keeping us well. Thanks. Yeah.
Dr. Vasoo, can I invite you to take a seat over at the sofas, and then we can conduct our Q&A from there. Okay, so this is a fascinating talk because you have a lot of questions to answer.
Oh dear, okay.
Okay, so I'm going to field, in fact, we have about 3 different people who have asked questions. Abhishek Rotre had asked, with regards to, I think, the DORMAT analysis where you compared the film array and the Verigene blood cultures. Can you shed some light on the false positives by that method?
As in the false positive results, or?
False positive results, yes.
So I think in terms of the DORMAT analysis is really how clinicians would behave when they're presented with a result. So in that study, if I do remember correctly, then that particular ranking system actually talks about the desirability of when you're presented with a result and how that impacts the antimicrobial choices. Certainly in a spectrum of antibiotics, I think in terms of greater and lesser evils, when you rank the antibiotics, some are obviously ideal. They're the most narrow spectrum, and also what will cause the least collateral damage in terms of when you use it and it's targeted to the organism. Then there's some antibiotics, because they do vary in spectrum, they are kind of intermediate, and then they are, and then those which are broad, while they might be microbiologically active, these would be inappropriate to use. And it's important to know that for the DORMAT analysis, when they do choose and rank, usually it's arbitrated by at least 2 physicians in the trial. And then you come together when designing the trial to agree on how you rank the antibiotics and contextualize it to that particular study. So in terms of the DORMAT, I don't think it specifically looks at basically false positives or negatives. It's more the desirability of how being presented with a rapid test result, how good it was in terms of guiding therapy and not, and how discriminatory it helped physicians to be in a real world setting. So it's a bit different probably from the traditional false positive, false negative approach. Yeah. But I think in that study, the accuracy actually was pretty high for the target included panel. So I think in general, the FDA approved panels really do have quite a high fidelity rate. Yeah.
Sure.
Yeah.
Thanks. So the second question also by Abhishek Rathre, it's do you advise testing positive blood cultures by MALDI-TOF, especially for bacteria and candida, to improve turnaround time?
Yeah, so I guess it's whether you are testing it from cultures. Yeah, I think certainly in this day and age, most diagnostic microlabs where you can, most labs have moved towards MALDI-TOF for organism identification. So I think without a shadow of a doubt, then for cultured isolates, MALDI-TOF is actually quite cost effective. Certainly the instrument itself is expensive. Sometimes maintaining the laser is, but certainly the cost per test is cost effective. I think the question is whether when you have a positive blood culture, or even before the blood culture flags positive, would you subject either a positive blood culture bottle, or if you go even upfront, blood directly from a patient's arm to a rapid test. So I think for that's what we are attempting to show here in the rabbit trial, that when you have a positive culture, I think at that time, if the clinicians a lot of times already before the blood cultures flag positive, depending on other biomarkers and the clinical condition and the history, may have already started empiric therapy. And we're trying to show that yes, it is worthwhile to, at least in some context, to have a rapid test to bring that more rapid identification. If it's gram positive and you work in a place where there's a lot of MRSA, for example, I think when I was working in Minnesota at Mayo, it seems like whenever there's a positive gram positive result, people will because MRSA rates are a bit higher there, and you don't want to miss that. And sometimes, even though they are contaminants, a lot of house staff, the junior doctors, would routinely start vancomycin. It's interesting, like in Tan Tock Seng and NCID, I noticed that people are not so, because we have lower MRSA rates, and because maybe it's a contaminant, people actually watch and wait a bit more. So I think even some subtle practice differences in different locales sometimes can be quite different. So if you work in a place where there's a lot more resistance, like a lot more community MRSA bacteremia, or for us, I think why we focus on the gram negatives in Singapore is that we do have a lot more resistant gram negatives. then I think implementing that test, such tests may be useful, and that's what we're trying to show.
Sure.
Yeah.
So you have 4 more questions. Did your study look at overall cost with the rapid ID ASP de-escalation strategy?
How are we gonna look at it? Yeah.
Yeah.
So cost is quite complex, and besides the, some folks are hospitalized for longer duration, and the bacteremic episode is just a single episode in their whole journey. So we are looking at cost and trying to break down the cost to the bacteremic episode and 2 weeks after the septic episode, because if you take the whole hospitalization, it may not be as accurate. And then of course we are looking at breakdown of costs with regards to antibiotics themselves. We did try to, as part of this study, to also demonstrate cost effectiveness. I think that's a piece where we are still cleaning up the data. We actually followed up the patients. We tried to, for a year, those which were not blood culture contaminants, and to administer a quality of life instrument to them. So to try to assess the cost effectiveness and any impact on the quality of life. So I think that's still work in progress, so I can't really fully comment on that as yet. Yeah.
Sure, we look forward to more of the results. Dr. James Sim had asked in Rabbit, does the lab do early MALDI identification on isolates, meaning the MALDI pinpoint colonies on agar plates after 4 to 6 hours of incubation instead of isolates after 18 to 24 hours of incubation?
Yes, so I think a lot we learned in the trial had to also do with workflow. For example, when you implement such a technology and the lab comes up with a really, really rapid result, do you have someone there in the middle of the night to really act on the results? So it's no point that we get all these fantastic results and no one acts on it and just sits there till the next day. So because this was a pragmatic trial, we did only run the trial during office hours, so like from 8 to about 5 p.m. And stewardship, all those results came in and we made those calls. If you were to ask me how in real life we might implement it if we were to introduce such a technology, I think you really do need then more real-time stewardship. I think in the middle of the night, people will be irritated if you called them to de-escalate, because they have a lot of other things to deal with. But certainly for escalation, I think people would do it because we all want to be right and save our patients' lives and make sure they are on the right antimicrobials. In terms of the bacterial testing then, yes, the lab did do MALDI on early growth. I think they would look at the plates like in the shift in the afternoon. If there's early pinpoint growth, the practice was to do an early MALDI on it. If not, then they would have to wait till the next morning. Yeah.
And Abhishek Ratri asked as well, does it give results on coexistence of the NDM and OXA48?
Yeah, I think for the ROSCO kit, yes, it would be expected to. I think we had isolates with multiple ESBLs. I don't recall isolates with multiple carbapentamases, but given that it is a chromogenic test, and it's actually agnostic. I think what is nice for phenotypic tests is that I think molecular tests will only detect the targets that you include on the panel. But if you have a good phenotypic test, then it is supposed to be agnostic to the beta-lactamase, and then it should detect the stronger carbapentamases. I think even for the substrate you use for the phenotypic test, it did have some difficulty. As you can tell, the OXA48, it was more weakly hydrolyzed, and some of the less common ESBLs, like some of the TAMs and shifts, the overall detection for ESBLs was about 70%. But for the CTXM group, which is the most predominant and preeminent ESBL in successful clones like ST131 E. coli, then it really didn't have a problem with that. Yeah.
Okay, last question before lunch, asked by Anirban Home Chaudhary. What are the causes of ambiguous results in the film array BCID?
Yeah, so I think the the results are not unambiguous because it's either detected or not, except that there were some instances, for example, where you need to correlate the gram stain with the readout. So at a later point in the trial, just for a short period, the manufacturer did issue an alert that there were reports of Proteus that was detected in the false positive results, and that really, I think, stemmed from nucleic acid material derived from the culture media. So I guess, and that was new to me in the trial, I didn't know that culture media, a lot of it might come from organic products with maybe plants, or I'm not sure plants, but the media that you use to make, there's different organic material, and so apparently it might sometimes have some extraneous DNA, and so you do really need to correlate. But other than that, I think that was mainly one of the issues that we found, but it was a small issue, and it was a time-limited issue. I think the other parts where it might be ambiguous is that you do get gram negatives, which are during the BCID1 panel, and not all gram negatives are on the panel. So if you get something like Chrysobacterium, for example, which we did have, and we also had myeloid in the blood culture, that's not going to in any way be reported. So it'll be gram negative seen, and we just call the team and we tell them that it's not on this panel, so we can tell you what we think it's not, but it could be anything else. Maybe so usually that leads to a phone call, and I tell them, and I look at the antibiotics, and this is going to be an unusual gram negative. It's like possibly a non-fermenter like Sternotrophomonas or something at that time which was not on the panel. And then of course I would be wrong because it's not Sternotrophomonas but Chrysobacterium glium. So but then we just tell them you need to sit tight and wait for the full culture results. So that is those are the limitations, I think, of any of the FDA technologies that we have showed, because even the Accelerate Pheno, for example, it's really what you include in the PNA fish probe, and you don't have something universal. So culture, I don't think culture is going to go away just yet, and it's going to be still the gold standard comparator.
Yeah, so absolutely right. All right, so thank you so much for sharing with us your fascinating talk, and I'm sure there's a huge amount of interest judging by the amount of question and answers. But so let's thank again Dr. Shawn Vasoo for this last symposium.
Thank you. Thanks, Catherine.
So.